So thanks to the organizers for giving us an opportunity to talk. It's always great to discuss with researchers in India about some of the developments that we're doing in Galaxy, and also see some researchers here. I can see Ravi Sardeshmukh, who's one of the attendees here. So what I'll do is I'll share my screen so I can present our talk on Galaxy P and how we use it for data science. So let me know if you can see the screen and I can start presenting then. All right, I hope you can see the screen. So basically, I'm Pratik Jagtap. I'm from University of Minnesota. I co-lead the Galaxy P team along with Professor Tim Griffin. And basically what I'll be talking today about is work that has been done by some of the researchers at the University of Minnesota. So we have a team of Galaxy P team wherein we have some developers from Minnesota Supercomputing Institute, as well as some of the researchers from our department heavily focused on mass spectrometry based data analysis. So in this talk today, I'm going to basically outline, give a little bit of introduction to Galaxy platform, and then talk about how we have added some proteomics tools into the Galaxy platform, and then give a little bit of introduction to mass spectrometry based research, though I don't know whether that would be required for this audience, but it's always good to at least give an idea about how we approach biomedical research. And then I'll introduce topics like proteogenomics and metaproteomics and what are the tools and workflows that we have used to extract information from the data that's available. I'll very quickly touch upon some of the research that we have been recently doing on mass spec data sets that are available in COVID-19 research field, and how reanalysis and newer insights actually are helping us to get an idea about how does the virus, what are the proteins that are present in virus, and some insights into co-infection studies as well. And lastly, I'll finish by talking about how can a researcher access some of the workflows and some of the tools that we have available in Galaxy. So in general, I think Anshu must have already maybe discussed this in her session today or yesterday. But Galaxy platform is a platform that is a web-based community platform. So basically, it gives an interface for researchers to use some software tools, and then you can not only use the software tools, but you can also use them as workflows. So you can actually generate a really large workflow wherein an output from one tool becomes an input for the next one, and you could process your data efficiently and in a reproducible manner. And you can also share these workflows. So it's very easy to take a data set, share your workflows using the Galaxy platform. And there are many public servers available all over the world, and you can access these servers. Either if you're based in Europe, you can get it through Galaxy Europe website, public site. If you're in Australia, you can access through Galaxy Australia. And if you're based in US, you can get through the Galaxy project, the galaxy.org site. So Galaxy, when it was developed for mostly genomics and transcriptomics tools. But in 2012, we basically started putting in some proteomics tools into Galaxy. And the idea there was not just to focus on single organism proteomics, but also try to look into emerging areas in proteomics. And the two areas that we kind of we identified were proteogenomics and metaproteomics, which have been kind of mentioned here. But I'll go a little more into details when I'm talking about them. So in general, what we start with is mass spectrometry data, and then you search your mass spectrometry data against a protein database, and then once you have your peptide spectral matches, you can either assign your peptides to as novel variants, or in a field which you call proteogenomics, or if you are searching it against a database which comes from microorganisms, you can also perform metaproteomics analysis, wherein you not only identify the function, the taxonomy of what are the different taxa that are present in your mass spectrometry data, but you can also perform functional analysis of the proteins that are expressed by these microorganisms. So I'll give a very brief introduction just so that there is some idea about how we approach research in general. So we basically start with proteins that are isolated from a sample, and it could be a sample from a single organism, it could be from human, it could be from a cell line, it could be from a soil sample, ocean sample. So there are various sources that we could start these to isolate these proteins from. These proteins are then digested with most of times with an enzyme called trypsin, and then they're subjected to liquid chromatography. The idea is to separate these peptides and then subject them to electrospray analysis. So basically what happens here is you ionize your peptides, and these ionized peptides are then separated based on their retention time in that liquid chromatography column, and as well as its mass to charge ratio. So that gives you an ability to get an idea about not only what's the mass of your ionized peptide, but also what are the different fragmentation ions that you get out of it. So the result of this is basically a spectrum so each peptide will give you a spectrum, and then using some data analysis methods or some search algorithms, you can assign a peptide sequence to each of the spectra. Not all of these spectra get assigned a peptide sequence, but most of them do. And that's what we use to basically assign these peptides to proteins, and you quantify proteins. And so there is more advanced analysis you can perform to get an idea about this. Again, this is a very general figure. There are variations of this, and you can use various search algorithms or various data analysis platforms to analyze your data. So I'll just basically introduce a little bit about proteogenomics. And again, most of you might actually be aware about this, but instead of searching against a standard database or a reference database, in proteogenomics, you end up searching against a database that's derived from either your RNA-seq data or a data which has got your variant peptides or variant protein sequences as well. So the idea here is to not only identify proteins that are present in your sample, which known proteins that are present in your sample, but also any variants because of any genomic variation that has occurred in your sample. And as a result of that, you end up getting proteins of various kinds that's been shown here. You have pseudogenes, you have protein coding genes, which are generally what are present in the reference proteome. But you also get proteins identified or peptides identified from your intergenic region. And so, and again, you could have novel splice isoforms. And so there are many ways that an organism can generate these peptides, and that's what proteogenomics ends up identifying. So what it basically does is proteogenomics, it confirms translation of variants. So if your RNA-seq data shows that there are variants present in your sample, your mass spectrometry data actually confirms that it shows that it's actually getting translated into protein. And then there is a direct evidence of potential functional variant. So you could have a protein which functions in a particular way, but maybe because of a mutation, a variant might function differently. Either in it might affect its catalytic activity, for example. And then it also has applications in neoantigen discovery, such as immuno-oncology. So what we ended up doing was, and that's what we have been working for the last few years, is developing a workflow that not only enables a search of these novel peptides, but also generation of database and confirmation and validation of peptides that are identified. So there is a pipeline that's available. In fact, there are 3 tutorials that are available on the Galaxy Training Network. So this is part of the Galaxy community, which wherein researchers have come together and decided, let's put in the most important workflows and most important tools onto this Galaxy site. So if you're interested in knowing more about this, you can go to this site, and I'll be very happy to share these slides after my talk is over. And then there are other workflows that are available as well in proteogenomics research that you could uh, actually not only learn about but also run them on the Galaxy EU server. So one of the aspects that we were interested in is that yes, we do have RNA-seq data and we do have proteomics data, and in both cases we also have quantitative information associated with that. How do we make use of it? How do we try to get new information out of this, rather than just sticking on to quantitative proteomics and then making biological conclusions from that, or just using um, quantitative transcriptomics and making biological conclusions from that, because there is a lot that one can learn, either getting some complementary information or even getting some information that might help, because of your transcriptomic information helps you to get the data. So Praveen Kumar, who was a PhD student here at the university, he developed a tool called Quantibe, and this was published last year in the JPR special issue of software tools. And basically what the tool does is it takes RNA-seq data along with the reference genome, performs alignment, there's a transcript of abundance pipeline. So there's a separate workflow which basically generates a transcriptome abundance report. And it also takes proteomics abundance report from another pipeline. And basically what the tool does is it looks at both of these, the transcriptomics quantitative data as well as um, quantitative proteomics data, and then tries to perform some statistical analysis to get an idea about what is the distribution of these abundances, and then perform some outlier analysis using some statistical tools. So what I'm going to do here is play a short video just to give you an idea um, about how this tool works. Um, so what you can see here is a Galaxy platform, and if I click onto the visualization sign, I can basically open this, and then on the left side here, you see transcriptome data and gives an idea about sample distribution. If I click onto correlation, it gives me an idea about how do these two data sets correlate, and then gives you various information about statistical analysis, regression analysis, and then also gives you an idea about what are the influential observations. So based on Cook's distance analysis, it tells you which of these proteins or which of these genes seem to have influence on the correlation analysis, and that actually gives you an important information about how these genes might actually be uh, influencing the correlation of these two. One other thing this tool also does is gives you an output about, a tabular output, so that one can look at these values and even filter them in case you wanted to perform any targeted analysis. It performs heat map cluster analysis, and eventually it also does cluster analysis of different groups, and you can determine the number of cluster groups that you want to look into. And again, all of these can be downloaded uh, in a tabular form if you want to perform some more analysis, or you want to use any of these terms for follow-up, either transcriptomics or proteomics-based targeted analysis. So that's one of the tools that we have. And then that's one of the tools that we have to get more information out of a proteo transcriptomics data. The other area that we work on is metaproteomics, and again, most of you might actually be aware of this. That microbiomes have been kind of a focus of research for last many years, especially with the fact that microbial proteins or genes seem to have an effect on response to stimulus. So for example, there has been a big focus on gut microbiome research, wherein it's been shown that your microbial composition can correlate with physiological conditions, starting from obesity to mental health to, many of these things. So it's getting importance not only in your health, but also your well-being, as well as in environmental research. And one of the areas that we are focusing on is, so most of the microbiome research uses metagenomics, so looking at the DNA that's isolated from either the clinical sample or from an environment, and then looking at the taxonomy of this. So there has been a big focus on making correlations based on this taxonomy, and there have been really sophisticated algorithms or methods available to analyze this. Metatranscriptomics is another research that has started emerging. So instead of just looking at the DNA that's present in a source of microbiome sample, you can look at what RNA is expressed by these samples. And here you get not only information about your taxonomy, but you also start getting some information about function, at least at the RNA level. What are the genes that are getting expressed in a particular condition? However, we have been working mostly because of the fact that we come from mass spectrometry background, on metaproteomics. But we are also interested in for one more reason. The metaproteomics actually gives you not only information about your taxonomy, which is what you would get with metagenomics and metatranscriptomics as well, but you actually get an information about the function. So you can look at what proteins are expressed, and from these proteins you can decipher which functions these proteins contribute to, and then you can find out what functions are getting expressed in a particular way. And so it's a more direct analysis, and perhaps a better way of understanding the mechanistic details of microbial interactions with either the host, if this is from a clinical sample, or with the environment, if it's from an environmental sample. So that's what we're working on. And what I'm going to basically show here is a data set that we have analyzed. So I'm just going to show an example of using some of the metaproteomics workflows. So this data set was from one of our collaborators in Norway. So this was from a food waste manure sample, and then this was basically grown at 55 degrees and then serially diluted and grown at 65 degrees in a laboratory condition. And then each of these samples was so this is basically has got cellulose that was added to these samples at various concentrations, and then it was also subjected to organisms that came from these food waste manure, and the idea was to see how these organisms degrade cellulose that's been introduced in these samples. And then the samples were collected at 3 different time points. We call it T4, T6, T7 has been shown here. And this comes from a lab from NMBU in Norway, from Magnus Arntzen. So this data has got spec data that was collected from these samples. So the question is simple here is, what are the organisms that seem to be present in the sample, and how do they degrade cellulose in this sample? So basically, what we did was we used this metaproteomics workflow, and I won't go into these details. I mean, I'll be very happy to answer any questions, or if there are any follow-up questions, I'll be happy to take them offline as well. So we have a workflow which performs peptide identification, peptide quantification, and also assigns function and taxonomy to this peptides that we have identified. And then we perform a statistical analysis by using a tool called as Metaquantum. So that's the last part that I'll be talking about. So based on this analysis, especially for this data set for T4, which is 23 hours, T6, 33 hours, and T7, we identified number of PSMs, peptides, GOTAMs, and genera that have been shown here. But this was just a list of peptides and its associated GOTAMs and quantification. So we wanted to basically go further and perform some quantitative analysis of this data, and that's where last year we published this tool called as Metaquantum. So what it basically does is it takes these peptides that are identified from your metaproteomic samples, and then there are tools that are used to assign MS1 intensity, so quantitative data, functional annotation, and taxonomic annotation. So there are these three features that are associated with these peptides, which we have already assigned using the earlier workflow that I talked about. But what Metaquantum does is, using R scripts that are present, that were used in this particular package, it generates, takes in this information and then generates information about data exploration, differential evidence, as well as cluster analysis. So this was one of the first few tools that actually can perform quantitative analysis on data of any kind. So you could actually compare two samples and come up with differential expression analysis. And I'll basically go through some of this, what we found out of this as well. So what's shown here is, so these are the T4, T6, T7 along with T1, which was also used as a data set. So T1 was the initial sample. And when we use taxonomy as an identifier for principal components analysis, we found that these data sets did not separate as well. However, when we use function, which is Go terms or gene ontology, molecular function terms, we found that the initial sample actually separated pretty well from rest of the samples, indicating that function is a pretty good identifier, or can be used as a really good feature to separate your samples. And that also gives us some confidence about why function in a metaproteomic sample is more important than looking at taxonomy. We also could generate some Volcano plots, and so what's shown here is T6 over T4 and T7 over T4. So these 2 time points, and there were some cellulose degradation proteins that were differentially expressed in these time points. And then we could also perform taxonomy analysis, so we could get an idea about what's the genus evidence. And we found that Hungarian, Clostridium, and Coprotomobacter are the most prominent organisms that are present in the sample. And we can perform heat map analysis from this data as well. And as you can see here as well, so what's shown here is in blue, red, and yellow are samples, T4, T6, and T7 samples. They are much better separated if you use function as a identifier or feature, as compared to taxonomy, where the separation is not as good, the one that you see on the right side. So one other thing we could also do with this Metaquantum analysis was taxonomy function interaction. And this is kind of an important part because we generally look at a microbiome as the functions that are expressed as functions that are expressed by the whole microbiome. But based on the fact that we have unique peptides coming from these data, you have an opportunity to ask questions like, for a particular organism, for example, Hungarian Clostridium, what are the different cellulose degradation enzymes, and how do they change? And you can see that the enzymes such as cellulase kind of increase at T6 and then decrease at 38 hours. And that's some kind of a pattern that we seem to see here. We can also ask another question saying, if there is a particular Go term or even a protein, what are the different organisms, how do they contribute to this? And you can see here, there are these 3 organisms which seem to contribute differently for glycoside hydrolase as well as glycosyl transferase. So it kind of helps you to get more deeper insights into your microbiome, telling you what are, informing you what are the different organisms that seem to be contributing to this microbiome, but also what are the different enzymes that are expressed by this microbiome. So lastly, I'll basically switch gears and talk about some really new research or new area that we have started looking into based on some of the workflows that we have already available. And we are basically looking at the large amount of data that is available or is going to be available through the COVID-19 research. And the Galaxy community basically took an initiative here and said, there's a lot of information available for COVID-19 research. Let's try to make the best out of this by using, or not the best out of it, but let's try to develop workflows that can either reanalyze this data or help us reanalyze any new data sets that come out of this, so that you can get newer insights into this. So there are some tools and workflows available on the Galaxy EU site, which for transcriptomics, genomics, uh, as well as proteomics. And what I'm going to talk a little bit about is the proteomics research that we have done. So as you're aware, there are quite a few clinical data sets that are available. So this particular figure is from a latest manuscript that came from that came out actually, I think just this month or last week. And it basically talks about various clinical data sets that are available. And it also, there are also data sets which give you an idea about the proteomes that are present in cell line model, as well as how does the COVID-19 virus interact with the host cells. And there is also a big push on targeted analysis. So the idea here is, once you have clinical samples, how do target your use targeted analysis to identify the viral proteins in the sample, and maybe use it as a way of diagnosis, either in terms of finding about the progression of the disease, or even finding out how does it interact with the host. So there's a lot of information available. So what we started with was these 5 data sets that I've shown here. So there's one data set from Armangod lab, which performs a deep proteomic analysis of COVID-19 samples in Vero cell lines. So the idea is to go deeper into the proteome. So they looked at a 4-hour time point sample, and then just try to find out information about this protein. And then there's also a time course analysis available from the same lab. And then there is proteogenomics data analysis from Matthew's lab. And again, this one is, all these 3 data sets are based on Vero cell lines. So you're not actually looking at human cells, you're looking at Vero cell lines to identify host proteins, in case you are interested in that. But there are also 2 of these data sets that came out recently about some gargling samples for COVID-19 infection. This came out in JPR from SINS lab. And there's another one which has got respiratory tract samples from, and then, so the idea for us was to go deeper into this and try to identify what are the different COVID peptides COVID-19 peptides that are identified from this. And so, as you can see here, we did a COVID-19 peptidomics analysis based on some of the workflow that we have. So we not only identified these peptides, but we also did a validation study using a tool called PepQuery, wherein you can go and interrogate your mass spec data and find out if that particular peptide is present in the sample with sufficient evidence. And then basically we came up with a list of these peptides that have been identified here. And we have a lot of information here. What we also found was shown in parentheses here are some peptides that were not earlier identified in these studies. So, and that was basically because of lack of availability of proteins of the right sequence. So there are certain peptides or certain proteins which are not annotated properly in COVID-19, or were not available in the public domain, but now they are. And basically because of our collaboration, we could get an access to these protein sequences, and we analyze this data, and we are waiting for the publication coming from these labs before we can reveal what these proteins are. But we have already found these some of these proteins which are novel variants of in COVID-19 viral samples. So that's an interesting place where we are hoping that we can get some information about what are the proteins or what are the peptides that are expressed or what are the proteins that are expressed by the COVID-19 samples. Another aspect that we have recently started working on is based on our metaproteomics workflows. We are also looking at some what are the different organisms that are present in a infection sample or a infected patient sample, apart from COVID-19, right? I mean, the idea is to obviously identify COVID-19 peptides in the sample, but also find out if there are any pathogenic organisms which could be co-infections, not necessarily primary infection, but co-infections that might lead to increase in severity of the disease. I mean, as you might be, I mean, as you I'm sure are aware that the COVID-19 infection leads to a spectrum of symptoms, right from absolutely asymptomatic to to eventual death. So there is a there are a range of symptoms that are identified, and one of the, I mean, obviously this can be attributed to your age, as well as your any predisposed conditions that you could have, pre-existing conditions that you could have. It also could be your immune status, but we also think looking at the co-infection status. So what are the different organisms or pathogenic organisms could also be an important thing, and that's getting some interest in the epidemiological field as well as microbiology field right now. So what we basically did was we used these data sets and use tools called as Compil 2.0, developed by some researchers at the Scripps Institute, wherein you can basically do a blind search of, you basically send your raw files, and it searches it against a really large database and gives you an idea about what organisms are present in the sample without you knowing anything about the composition of this sample. So you don't need a protein database as such. You basically searching against the large database there. And what we found was by using UniPep and PepQuery that there are peptides of validated peptides that of from these organisms that are present, and some of these are all of these organisms are actually either nosocomial infections, so infections that occur in hospital, or could have some effects because of its antibiotic resistance. So that's something that definitely one should look into for these samples. We also looked at gargling samples, so samples that were collected from gargling solutions from 3 patients, and we found 2 organisms, Stenotrophomonas maltophilia, which has been known to be an organism that's present in your oral cavity or in your throat, but it has shown to have some sometimes can be pathogenic as well. And Streptococcus pneumoniae has been known to be a pathogen that's been depending upon the condition can affect your how your disease progresses. So we are looking into this, and we are soon going to kind of publish this. I mean, if you want more information, there is if you go to this site that has been shown here, you will get more information about all of these data sets and reanalysis that we have been doing here. So with that, I'll come to the end of the talk. I hope I'm doing all right with my time. So based on so there are various ways you can access these workflows. There is there is a GitHub site, there is Galaxy Toolshed, wherein we have all the tools. There are training workflows available on this site here. Our published manuscripts are accessible through this link. We also have presentations available on this website. If you're interested in collaborating with us or want to know more about our work, you can always contact us here, also on my email address, and I'll be happy to share that when I share the slides. We also are on Twitter, so if you want to share, reach us through that, we'll be very happy to communicate with you. Lastly, I would like to just acknowledge that we not only work with research at the University of Minnesota, shown some of the biological researchers, as well as the Galaxy P team and the Minnesota Supercomputing Institute, but we also collaborate with researchers all around the world, especially for either software development or implementation, or even working with some of the research projects that we work on. With that, I'll be happy to take any questions if I have any time. Otherwise, I'll be happy to answer them via email. Thank you.